All right, so we talked about this idea of lactate threshold being the result of an increase in production, but also a decrease in lactate clearance. And that's why I gave you the Stanley paper that I gave you. This paper, I really want you to appreciate what this paper is. This is not this is not a simple experiment that it should be kind of discarded. The 6 individuals who took part in this study, they actually had to sign a waiver that said they wouldn't have children as a result of the fact that this is actually using C14 radioactive carbon. So it's lactate that's been tagged with a radioactive carbon. And so what they've done here is they've given humans lactate with this carbon 14 tag on it. And what it allows them to do is it allows them to track the fate of the lactate. They can then calculate from how much C14 lactate they've done, given, they can calculate the rate at which lactate is appearing in the blood, new lactates being produced, versus the rate of disappearance, the rate at which lactate is being used in the blood. And then they can calculate they can calculate from this data the maximal clearance rate. So this is how much the lactate is being cleared. And so now what we can get is we can take a look at the classic study that says, here's lactate. It goes up linearly as we increase exercise intensity, but then at a certain point, we leave the linear relationship and we go through, and there's really a couple of lactate thresholds. You can see that there's one here where at 14 minutes, they start to go up off of that linear increase. And then there's one where it becomes really, the slope increases really, really steeply. And so classically, these are 2 different lactate thresholds. And if you get into training athletes, that's something that you'll have to learn a little bit more about, how these 2 different lactate thresholds are really slightly different states at which we're really competing in. And what you can see is this is what we'd see in a normal lactate threshold test. You're doing an increasing exercise, you take blood pops, you put it onto your lactate meter and you read your lactate, and you get that every 2 minutes and you see what your lactate is. But it doesn't tell you whether it's from producing new lactate or whether it's from decreased clearance. And now what Stanley did by putting in this C14 labeled lactate is now you can follow the lactate. And you can see if I put in this amount of lactate, there should be that amount of lactate. And now if I see that the lactate concentration actually goes down in the blood, my C14 lactate goes down in the blood, but my total lactate concentration stays the same, that means I've produced more lactate. And if it goes the opposite direction, I've reduced or I've disposed of some lactate. So let's take a look at now, how does this work? We see the first threshold is happening about 14, and then the second one happens about 20. So now let's look at rate of disappearance. Rate of disappearance is going up in a linear fashion, and it stays linear out to 16. Okay, but we saw that the rate of lactate accumulation in the blood, so our bank account, is showing that it's at 14 we're seeing a threshold. So you can see here that lactate production or lactate appearance goes up at 14 minutes, or right before 14 minutes. And this is probably why we see this first uptick, is that, yeah, there's a little bit greater production, but the appearance is staying relatively decent. The disappearance is tracking fairly well. And you can see that as lactate goes up, the rate of disappearance actually goes up right after that. So it's a little bit of a lag period. So this first time, we're not really seeing anything other than the rate of clearance is lagging a little bit behind the production. But we're still clearing the majority of the lactate, and you can see that we're clearing out the stuff that we were accumulating here. So now what happens is we get up towards that 20-minute point where we're seeing that secondary peak. What you can see is that now the rate of appearance is far outstripping the metabolic clearance rate. You can see the metabolic clearance rate here, and that is showing us that at 14 minutes, we're getting up towards a plateau. So we're no longer able to increase metabolic clearance of the lactate. And what we're seeing is that's actually coming down from the second part of the test. So we're producing far more than we can clear, and that's what we're seeing as this accumulation. And so really what we can see here is that, yes, we can increase clearance a little bit, but really the production rate is going up much more. And then if anything, our clearance rate, our ability to clear what we're producing is going down. And so now the question is, why is it going down? So, and this study, just so that we're clear, was done at UC Berkeley by the father of lactate research, and that's George Brooks. And George Brooks is a mentor of mine. He's an incredible scientist, and he's really been the father of lactate research because he was a national level runner who was told that the reason that he wasn't an international level runner is because he was producing too much lactate. And that was going to be the problem, was that he just didn't get enough oxygen delivery. And so he basically spent his whole career, and he's continued to work till today, and he's spent his whole career trying to figure out how lactate is related to performance. Okay, so this first study showed us that we're producing more lactate than we can clear, but our clearance rate, and we're producing lactate even when we're going at low intensity. Even when our lactate is super low, we're still producing a good amount of lactate. We're producing 50 micromole per kilogram per minute. That's a lot of lactate, but we're just clearing it all. And so it's not necessarily that we're producing a lot of lactate. It's how are we getting rid of the lactate we are producing. So this is a follow-up study by Lorraine Turcotte still in George Brooks' lab. And what she did in this study, she's now at the University of Southern California. What she did in this study is she used a drug called MPA. And this is an inhibitor of PepsiK, which is an enzyme necessary for gluconeogenesis. And what she did is she measured glucose levels in rats that were exercised for, and you can see, for up to 120 minutes. And what they looked at here is they looked to say, okay, the exercise was from 60 minutes to 120 minutes. Apologies. And so what we want to do is we want to look at what the glucose level in the blood is with exercise. And this is not a high intensity exercise, but they start them running and they get them going, and they see that the control animals, the glucose levels stay normal, and they stay at this kind of 7 millimolar level. Now let's look at what happens if you block gluconeogenesis. Now we're looking at blocking gluconeogenesis. What happens to blood glucose? First of all, it starts lower. At rest, you're using gluconeogenesis, where you're converting 3-carbon sugars into glucose as a way to maintain your blood glucose. Now we start to exercise. Now, even low-intensity exercise where our blood glucose is good, it's going up, if anything, in the controls. It's going down in these animals that have the MPA treatment. This is this inhibitor of PepsiK. So what we're seeing is we're decreasing blood glucose by having this inhibitor of gluconeogenesis. And now what you're going to do is what we can do is we can take a look and see how much glycogen there is in these animals. So in the liver, sham injected, so these are the controls. You can see that they've broken down about 180 units of glycogen, and this is micromole per gram of tissue. Now let's look at the MPA injected animals. Blood glucose is dropping, so maybe we're blocking liver glucose production or liver glucose breakdown. You go from 240 at rest down to 13. So these animals are going almost to no glycogen in their muscle. You can see the same thing is happening in the working muscles. Normally, they would use about a third of the glycogen that they have in the muscle, but here now in the MPA treated animals, blood glucose is going down. They're having to use more of their stored glycogen, so they're using 2/3 of their glycogen in their muscle instead of using about a third. So now you've had to use twice as much glycogen in your muscle because you can't make glucose in your liver and your kidneys, and you've depleted almost the whole of your liver glycogen. So what this is telling us is that in steady state exercise, where we're normally just going along, there's a lot of glucose production using 3-carbon sugars, using maybe some amino acids, but it's mostly coming from lactate being converted back into glucose. So what happens if you instead increase lactate? And so this is again done out of George Brooks's lab, and this is by Ben Miller, who's now at the University of Oklahoma. And what he did is he had people exercise at 65% of their VO2 max, 55% of their VO2 max, and you can see that the lactate in the blood was lower here. But then they had a second group where they exercised at 55% of their VO2 max, but then they added lactate to make up for the difference in the amount of lactate that was in the blood. Because at 65%, these individuals were above their lactate threshold. At 55%, they were below. So the difference in lactate was actually pretty big. So what they did is they added that amount of lactate in to see what would happen. The first thing that happened is nobody felt that their muscles got more sore, and their muscles didn't start to ache more and feel, oh, I'm feeling burning in my muscle. Not at all. Okay? How much muscle glycogen did they use? Exactly the same amount. How much lactate did they use as a fuel? Look at that. The extra lactate that you added actually just meant that you used more lactate as a fuel. So what happened so you used the same amount of glycogen in your muscle. Now you're using a lot more lactate as a fuel. What happened what's the difference? Well, you actually decrease the amount of blood glucose you use. So that means you're protecting your liver glycogen so that you're not oxidizing as much glucose from the blood. That means your liver doesn't have to produce as much. Because now instead of using liver glycogen breakdown to produce glucose, you can use this lactate and either go through gluconeogenesis, or you can just use it as a fuel for your muscle cells. All right? So how much gluconeogenesis do you have? Basically, this is liver gluconeogenesis. This is glycolysis. So are you using the lactate to produce more glucose, or are you oxidizing it directly? And so what you can see is 55% here means that you're using glycogen breakdown. So you're breaking down glycogen in the liver. And so this is liver glycogen breakdown here. And then what you can see at 55% is that liver glycogen breakdown has gone way, way down. And now instead of liver glycogen breakdown, now the fate of the lactate is to go into liver gluconeogenesis. So what happens to lactate is that we don't use it, we can use it as a fuel in our muscles, but we also are using it and we're reconverting into glucose. And by reconverting into glucose, that means we can spare liver glycogen so that we can use that liver glycogen at much higher intensities. Okay, so normally what you're doing at a steady state exercise, you're producing lots of lactate, but that lactate is being taken into the liver and the kidneys and is reconverted into glucose. And that means that you don't have to use your liver glycogen to keep blood glucose levels normal. All right, and so this is really one of the key fates for lactate, is it goes out and it can be used as a fuel throughout our body. It can spare liver glycogen and it can be used directly as a fuel.